Hello, everybody. I'm Anthony Felix, and Ethan and I chose our topic, DNA and RNA nucleotides, for this project. The difference between DNA and RNA nucleotides. DNA and RNA nucleotides differ according to which 5-carbon sugar is present. They also differ according to whether the nitrogenous base thymine or uracil is present. DNA contains a deoxyribose sugar, while RNA contains a ribose sugar. DNA contains the thymine nitrogenous base, while RNA contains a uracil nitrogenous base. What makes up these nucleotides? For DNA, each nucleotide in DNA contains one of 4 possible nitrogenous bases. It can contain adenine, guanine, cytosine, and thymine, and their respective abbreviations are there. Adenine is A, guanine is G, cytosine is C, and thymine is T. For RNA nucleotides may also contain the nitrogenous bases adenine, guanine, and cytosine, but instead of thymine, RNA replaces it with a different base called uracil, and uracil's respective abbreviation is U. Functions of both DNA and RNA. DNA replicates and stores genetic information. DNA is also a blueprint for all genetic information contained within an organism. For RNA converts the genetic information contained within DNA to a format used to build proteins, and then it moves it to ribosomal protein factories. Comparing the lengths of DNA and RNA. DNA is a much longer polymer than RNA. A chromosome, for example, is a long DNA molecule, which would be several centimeters in length if completely unraveled. On the other hand, RNA molecules are variable in length, but are much shorter than their DNA polymer counterparts. Where are these nucleotides found? DNA is mostly found in the nucleus, but DNA can also be found in the mitochondria in only small amounts. RNA forms in the nucleolus, however, and RNA then proceeds to move to specialized regions of the cytoplasm, depending on which type of RNA is formed. And that's my part. I'll pass it off to Ethan.
There are 3 main parts to nucleotides. There's the phosphate group, which is a phosphorus atom bound to 3 oxygens and an alcohol. The second group is sugars, which is a pentose sugar in all nucleotides, which is 5 carbons. Deoxyribose sugar is for DNA, where ribose sugar is for RNA. Deoxyribose has one less sugar, where ribose contains another sugar. The third and final group is the nitrogen bases, which is thymine, cytosine, adenine, guanine, and uracil. There's 2 main groups of the nitrogenous bases. There's the purines and the pyrimidines. Within the purines, there's adenine and guanine. Both these contain 5 carbons and 4 nitrogens within their 2 fused rings. The pyrimidines are cytosine, thymine, and uracil, which are all single-ringed. The way nucleotides bond to one another within a DNA structure is a purine to a pyrimidine. Adenine bonds to thymine with 2 hydrogen bonds, guanine bonds to cytosine with 3 hydrogen bonds, and in RNA, where there is no thymine, the adenine forms 2 hydrogen bonds with uracil.